Thank you everyone for joining me today. My name is Amanda Grimm. I'm a Senior Segment Marketing Manager here at GenScript, and my presentation is Precision Diagnostics Accelerated, GenScript's portfolio for modern IVD innovation. I'm excited to share with you today all of our cutting-edge technologies to help you in your diagnostic development. The contents of my presentation will include some information about GenScript. I'll touch on some innovation in vitro diagnostics. I'll go then into our solutions. I'll highlight a couple customers, case studies, and then I'll close out with some information on partnering with GenScript. All right, we'll start with some information about GenScript. If you're new to learning about GenScript, we do have over 20 years of development experience, having started in gene synthesis. We're always at the forefront to provide the newest technology in order to improve our methods as well as our output. Our CRO and CDMO foundation provides us with a range of experience, having multiple partners in therapeutic spaces and working with them to bring their antibody drug, cell or gene therapy, vaccines, or diagnostics to market. And then because of COVID, we were able to offer the first of its kind neutralizing antibody detection kit across multiple countries and regulatory bodies. So that adds a lot to our regulatory experience as well. We were founded in 2002 in Piscataway, New Jersey, and since that time, in addition to gene synthesis, we've grown our molecular biology offerings. We've also added antibody and protein expression services, peptide synthesis services, and even instrumentation. We've also expanded the GenScript umbrella to include our synthetic biology product segment, BestSign, Legend Biotech, which is our cell therapy segment, and then our CDMO ProBio. One thing I do want to highlight is our partnership that is between Legend and Janssen to work on a cell therapy product, and they did receive regulatory approval for that in 2022. We have already updated and launched new products this year, going into 2025. So I encourage you to follow us on social media, check out our websites to make sure that you stay up to date with everything that we have going on. Here we have a further breakdown of each of the subsidiaries. Like say I'm with the life science CRO, and then we have our CDMO, our cell therapy, and our synthetic biology subsidiaries. All right, let's go ahead and move into the innovations in IVDs and their contributions to precision medicine. Start with companion diagnostics. These are diagnostics that detect a biomarker associated with a given therapeutic. Biomarkers are not specific to a single indication and can be cell surface proteins or genetic mutations. The therapeutic itself can target multiple indications. An example is the antibody drug pembrolizumab, that this inhibits PD-1. PD-1 is the programmed cell death protein 1 found on T cells. It's an immune checkpoint protein, and its receptor PDL1 is expressed by multiple normal tissue types, but is overexpressed in many cancers as a way to circumvent our immune system. Companion diagnostics are best developed in a co-development strategy with new therapeutics. Their incorporation can ensure a new therapeutic is specific and targeted, and can also prove efficacy by identifying individuals who will actually benefit the most from receiving treatment. First companion diagnostic was approved in 1998. This was an immunohistochemistry test for Herceptin, identifying the overexpression of HER2 in breast tissue. Since then, we've also seen new technologies be approved as companion diagnostics. That includes in situ hybridization, in this case using fluorescence, that's the FISH abbreviation. This uses fluorescent DNA probes in order to bind the target nucleic acid. We also have RT-PCR for the BRAF V600 mutation for melanoma treatment. We also see MRI imaging in the aid in treatment in order to monitor liver iron concentrations. And then we also see next-gen sequencing in order to provide a qualitative detection of BRCA1 and BRCA2, looking at specific gene alterations. And then a newer type of assay that was approved in 2023 is electrochemiluminescence immunoassay. This looks for the selection of patients that would benefit for a treatment for adult hemophilia A. Both PCR and NGS have revolutionized companion diagnostics by enabling the precise identification of genetic mutations, leading to more effective and personalized treatments. From a regulatory standpoint, the safety and effectiveness of these technologies have been critical for regulatory ensuring that the companion diagnostic tests meet high standards for clinical use. I'll talk more about NGS and introduce CRISPR-Cas in the next couple slides. Gen sequencing has emerged as a transformative tool due to the ability to analyze multiple genes at once with high sensitivity and specificity. It's shown to be highly effective to support personalized treatment selection in complex diseases like cancer. It has a range of benefits that include advanced insights when compared to IHC, which was limited to single targets. The sample source flexibility that it can be applied to formalin-fixed, paraffin-embedded tissue or minimally invasive liquid biopsies. This then aids further patient monitoring downstream. It also addresses a range of therapeutic applications in oncology, so by determining mutations of the EGFR or even BRCA1-2 and more to guide those therapy decisions. It also addresses inherited disorders to help match rare genetic conditions to those targeted therapies. And then, of course, immunotherapy, so biomarkers like TMB and MSI are going to be examples of emerging for predicting responses to checkpoint inhibitors. Most familiar with CRISPR's success as incorporation into COVID-19 point-of-care diagnostics. The Cas12 and 13 based systems like Sherlock and Detector allow for rapid, low-cost, and sensitive nucleic acid detection, paving the way for point-of-care companion diagnostics that could match patients with therapies more efficiently. Additional applications include mutation detection, so detecting EGFR mutations in small or in non-small lung cancer patients in order to determine suitability for those tyrosine kinase inhibitors. We also have gene expression profiling, so CRISPR-based assays being adapted to detect RNA expression levels. This is helpful in assessing biomarker profiles that are predictive of drug efficacy or even resistance. In this next section, I want to introduce GenScript's solutions to support your companion diagnostics and next-gen molecular assay development. Here we have a high-level overview of the development process. This starts with identifying your disease of interest, going through target discovery to identify whether you'll be looking at protein, antibody, or nucleic acid. Then you have designing and optimizing your assay, moving into validating and verifying through a number of procedures of data analysis, sample collection, and patient testing. And then after submission and approval, it can now be used for diagnostic use. From the development workflow, we have 3 stages we've identified where we can provide solutions. We have target identification, assay format and validation, and then assay production and quality control. GenScript's offerings align with 2 diagnostic formats, the first being antibody or antigen detection, and then the second one being molecular detection. Part of understanding the target is getting to the root of the biology and mechanism of the disease or condition involved. The information uncovered leads to not just the therapeutic target, but the diagnostic that supports the personalized medicine to ensure the right people receive treatment. Target identification may involve a number of methodologies in order to confirm target functionality related to the disease. So this section is divided into 2 areas of application: gene analysis and protein analysis. GenScript's GeneTitan is a semiconductor platform that can synthesize 8 million individual oligos on a chip. By combining either hybridization or amplicon-based enrichments, this technology provides insights into various genes and their function. GeneTitan can also be used to create customized gRNA libraries that are cloned into dual or all-in-one CRISPR vectors that will give additional methods in order to study your gene functionality. We also have ready-to-use guide RNA libraries as well for commonly investigated diseases like Alzheimer's and diabetes. CRISPR nucleases play a critical role in gene editing and can help you create a model to better understand the intricacies of disease and the potential for protein-protein binding. We offer the variety of off-the-shelf nucleases such as Cas9, 12A, 13A, and prime editors to customize your site-specific gene editing strategy with the incorporation of our single guide RNA service. This is going to allow you to create a couple of targeted edits or build your own library that's going to offer an alternative method for high throughput screening of oncogenes or tumor suppressors across multiple cancer models. We can also assist with the right delivery methods, including lentiviral vectors that will allow the single guide RNA to integrate into the targeted cell. To continue the evaluation of target genes and functionality, we have a comprehensive range of ready-to-use PCR and qPCR reagents, as well as complementary custom services. Our qPCR and PCR ready-to-use reagents are formulated to provide reliable amplification of target DNA or RNA sequences, resulting in sensitive and specific detection. Our custom oligo services includes primer and probe design, optimization, and synthesis. This is going to ensure successful identification of specific targets and biomarkers, and these probes and primers can be provided in a tube or even a plate for high throughput testing. Antibodies provide an integral role in development where they can function in target characterization, antigen capture, or antigen detection. We do offer a wide range of catalog antibodies that can be purchased and are ready to use. This range includes anti-VHH, epitope tag, anti-idiotype, and immunodetection. We also have a large catalog of IVD antibodies that includes antibodies and antibody pairs for conditions like bone metabolism, fertility and pregnancy, and that are also tumor specific. These antibodies have been characterized and indicate specific use applications that they've been tested for, like ELISA or flow cytometry. GenScript has a range of antibody services that can be fully customized. This includes the type of immunogen that you're using, whether it's DNA, mRNA, or peptides. We also offer our proprietary immunization technology in order to expedite the timeline. Our platforms include polyclonal, traditional rodent hybridoma, and we also have our monorab rabbit platform. The rabbit immune system offers a significant advantage due to its unique immune system capability to achieve highly sensitive and specific antibodies with little to no cross-reactivity. Our products division offers a comprehensive suite of instruments and reagents for Western blot automation to support protein analysis and characterization. Once expressed and purified, your proteins can be checked for size confirmation and denatured or native conformation reactions via downstream testing. The precast gels are available in various formats and percentages, so you can choose the most suitable gel for the specific experiment and its requirements. In addition to gels, we also offer equipment to support each stage of the blotting process, from transfer systems to blotting to staining. And then to complement the gels and equipment, we also provide an extensive collection of catalog elite antibodies to assist with that protein analysis. They're used for epitope mapping, antibody validation, target antigens, or as antigens themselves for antibody production. We have a catalog of ready-to-use peptides that cover diabetes and obesity, Alzheimer's, cancers, and even some libraries. If you need to customize your peptide, we recently launched our TurboTide service to expedite that synthesis and delivery to you. Additional options include support with peptide design, modification, and labeling options to meet your specific project needs. If you have a protein or your biomarker that needs to be expressed, we have a comprehensive protein expression service that covers multiple expression platforms to accommodate the diverse needs and to optimize and scale the production. The mammalian expression platform offers the advantage of post-translational modifications and native folding, making it suitable for the production of complex proteins and antibodies that require proper folding and glycosylation. The system is going to ensure the generation of biologically active proteins that will closely resemble their native counterparts. The insect expression platform offers an alternative for producing challenging proteins that require proper folding and also post-translational modification. It's an alternative method to produce recombinant proteins with high yield as well as functionality. For more simple protein expression needs, we have the E. coli platform. This provides a cost-effective and rapid solution. It's going to be ideal for the expression of soluble proteins, protein fragments, or small-scale production of antibodies for initial screening and characterization. Newly added to our protein expression services is the cell-free platform. This does use E. coli cellular machinery, but without the cell component. Among our mammalian expression platform is our proprietary CHO cell line, the TurboCHO platform. This is going to be a scalable platform for recombinant protein and antibody expression. So as you move through the process and the development, it's going to scale with you. We have the high throughput in the discovery stage, which is the transient expression, providing a fast turnaround at 5 business days, moving into the express as protein amounts those protein needs increase. It's also transient, but can also support many different antibody formats if that's needed as well, and then to high performance and stable pools in order to support later stages of development where large stable quantities are needed for clinical studies and commercialization. Okay, now let's take a look at the next stage of development, the assay format and validation stage. Common assay formats here are going to be like the ELISA, lateral flow, or as we saw early on, this would include even immunohistochemistry. We also have PCR, qPCR as well. Assay development considerations include relevant controls, conditions, and reagents necessary for the test to be performed. Once the design is complete, conditions need to be optimized in order to ensure that the assay is sensitive, specific, and reproducible. This next section, as I go through, I'll first talk about solutions for antibody and antigen detection, and then I'll move into molecular detection development. Okay, starting with antibody and antigen detection solutions. In the previous section for target identification, I talked about our different antibody services. So this is a place where you can optimize that antibody further. Precision mutant libraries and our mutation services are great tools to facilitate the development of more accurate and sensitive diagnostic assays. So the precision mutant libraries consist of a collection of systematically designed mutants that enable you to explore the functional significance of specific mutations in their target proteins. And then our mutation services allow for the generation of custom designed mutants, providing flexibility in studying and optimizing antibodies. Mutation options include site-directed saturation scanning and combinatorial mutagenesis. Performing optimization and validation testing is going to require large volumes of antibody. So you want to make sure there's going to be enough in order to fine-tune the assay for the optimal performance, the accuracy and reliability without having to constantly make more. It's also an important part of the scale-up process for your assay materials. We can offer both in vivo ascites or in vitro, such as roller bottle scale-up of the hybridoma production. I've broken this into 2 slides in order to simplify the info, but here we see the ascites production workflow, and that yields 2 to 5 mils of 1 to 10 mg per mil per mouse. And then in the second slide, it shows the hybridoma workflow where we can go one of 2 routes. The first being hybridoma expression across the top there, yielding 15 to 50 milligrams per mil, starting at a single liter production volume. Or the second is sequencing the hybridoma antibody to generate a recombinant that can then help to further increase yields over 100 milligrams per liter in volumes ranging from 100 mils to 200 liters. Antibodies or proteins may require labeling with a fluorescent dye or enzyme like horseradish peroxidase. If we think back to the beginning, I talked about the IHC Hercept test. This would be an antibody that uses an enzymatic reaction to generate a color. We can also do fluorescent label for flow cytometry. Antibodies are also commonly used in ELISAs, but these are options part of our antibody reagent modification and purification services. Before we move into the molecular section, I want to visit the IVD antibodies that we have that are ready to use that I mentioned earlier during target identification. We have a range of these that address many of the commonly investigated health conditions and diseases, like the bone metabolism as well as Alzheimer's. These antibodies are produced with high quality standards and have superior lot-to-lot consistency. They are also highly characterized, so we know their isotype, their working format, like immunofluorescence, ELISA, etc., as well as recommended antibody pairs. These are ready to use and can be purchased in large quantities. Okay, let's transition from antibody and antigen over to molecular detection assay development. I talked about the emergence of CRISPR-based diagnostics for viral detection, and that being quite successful in the case of SARS-CoV-2 as well as some other viruses. Adding CRISPR-Cas offers flexibility, whether you're developing a fluoro-based molecular detection system or taking lateral flow to the next step. The pros of this technology is that it can help lower associated costs compared with traditional PCR, and by doing so opens up access to identify people for novel therapeutics that may have cost barriers involved. The diagram here is from the Kaminsky publication. It outlines those different pre-amplification pathways and different associated Cas enzyme used to develop different types of diagnostics. GenScript does have those ready-to-use Cas nucleases that cover each of the Cas9, 12A, and 13. These can then be combined with the customized single guide RNA service. We do offer a number of catalog product reagents for PCR and qPCR. This includes the enzymes, dNTPs, and plasmid vectors. When you couple that with the oligosynthesis service, you end up with primer and probe product with low contamination, highly precise and sensitive, consistent labeling, and a very stable product. Last stage, we're going to move to the assay production and quality control. So here, the validation of the diagnostic assay adheres to the processes outlined by local regulations. Here in the United States, the 510(k) trial and the de novo, these are established by the FDA, and it involves collecting sample data for analysis. That analysis evaluates the precision, accuracy, reportable range, reference range, analytical sensitivity, specificity of the assay. In order to maintain accuracy and reliability over time, quality control procedures are implemented early on and carried through to commercialization. To preface, this section relates to ISO production, as this may be a customer request of the product that we provide them. We can also elevate the quality standard of other materials not listed here. We do want to assure that the raw materials used in your diagnostic assays are of the highest quality, meet your regulatory requirements, and contribute to the accuracy and reliability of that final diagnostic product. So again, we'll separate this section into antibody and antigen detection, and then we'll look at molecular next. For high quality recombinant antibody or protein production, we have the high performance option from the TurboCHO platform. It's a key option for expression of high amounts of protein or antibody from 500 milligrams to kilogram levels. This can be done in as little as 6 weeks in order to express the recombinant proteins or antibodies. The QC testing is standard, and it includes SDS page and endotoxin control. We do have additional options like FACS or SPR that you can request as well. Here I'm revisiting the large-scale antibody manufacturing services that I mentioned in the last section. Again, we offer scale-up production of monoclonal and polyclonal antibodies from that milligram to gram level. Each of these is going to be available with mycoplasma testing, mycoplasma removal, as well as clonality services for the highest quality antibody production. Right, next we'll look at molecular detection. Talked about CRISPR and the Cas enzymes earlier being incorporated into diagnostics. We do already offer GMP CRISPR capabilities for therapeutic materials, so we're able to take advantage of this and be able to adapt with the QMS level production that you need for your assay. The last one to share with you is the oligo service We're able to provide this as well at an ISO quality level with deliverables, including a C of A and MS report. And then here in this next section, I want to take a moment to highlight a couple of our customers and their work. For the first customer highlight, the goal of the study was to develop a robust and efficient method for identifying Alternaria species using analysis of the whole genome. This method aimed to facilitate a timely detection of the species, enabling targeted treatments and mitigating associated damages. Ultimately, the outcome was that they successfully constructed a species-specific target sequence library of 31 of the Alternaria species. The method was validated using Sanger sequencing and CRISPR-Cas12a technology. The service that GenScript provided in the study was to provide the synthetic CRRNAs for the CRISPR Cas12a system. These were crucial for the rapid and precise identification of the genomic DNA samples. The CRRNAs were synthesized to target species-specific sequences, enabling that detection of the Alternaria species with high specificity and sensitivity. And for the second customer highlight, we have a group that the goal was to study and investigate how the SARS-CoV-2 ORF3a protein induced apoptosis through mitochondrial dysfunction, specifically focusing on the role of the potassium ion channel. The outcome was that they found that this triggered significant mitochondrial damage, leading to that apoptosis and inflammation in lung tissue. The addition of exogenous potassium was shown to mitigate these effects, suggesting a potential therapeutic strategy to reduce severe complications associated with COVID-19. And the GenScript service that they used for this was using us to generate the full-length DNA fragments of the SARS-CoV-2 ORF3a genes. These fragments were used in the study and enabled the researchers to express the ORF3a protein in HeLa cells and mice models that facilitated the investigation of its role in that apoptosis and mitochondrial dysfunction. Last section, I just wanted to share some information on partnering with GenScript. Latest data primarily from the end of June 2022, so I definitely know that we've exceeded these numbers. And I do want to note that we've been cited more than 100,000 peer-reviewed journals, and as indicated in the graph below, this number continues to grow. And we want to continue to grow and support all the important research being conducted. A summary of what it looks like to work with us. It can be as simple as some plasmid work, or if you wanted a more integrated workflow. Our teams love to provide support, and we hear that feedback often where customers are happy with product deliveries and they never have to question what's happening, since our project managers are amazing in order to keep you up to date on your projects. I just want to say thank you for your time and attention. Please visit our virtual booth. We have a number of resources, whether it's literature or additional video links that may help you in your research. Please feel free to reach out to me or your local sales rep. We'd be happy to help get you going and answer your questions. Thanks.